Fantastic. Good afternoon. I hope we will have a nice and sparkling conversation and debate for you, being a late afternoon on a Friday. My name is Jane Metcalf, and I'm the founder of NeoLife, which is a newsletter and website and a book about how our technologies are enabling us to transform our own biology, also known as the co-founder of Wired. Our session this afternoon is called Negotiating the Boundaries of Our Genetic Future, and we have an amazing panel of people assembled here to address that. Of course, the cost of sequencing has dropped from $3 billion to, sometimes you can get it for less than $100. We can not only read our DNA, we can edit it and we can write it from scratch. We can use these technologies to prevent inherited diseases and cancers and cure them. And in another 25 years, as you've just seen, we could perhaps be using this technology to augment our senses and allow us to travel and survive and even thrive in space. It was just a few short years ago that He Jiankui has edited the genome of these two twins, Lulu and Nana. Where are they now? What is their health like now? Are we now on a course to a genetic determinism, which is demonstrated by movies like Gattaca? Are we looking at a Black Mirror future where our genetic fingerprints are used to control us? Is this the future that science fiction has prepared us for? And if so, how do we balance that with the opportunity to cure and prevent disease and extend our species into outer space and all the other promises that go along with that? We are incredibly fortunate to have George Church with us today, joining us from Cambridge, Massachusetts. He's probably needs no introduction, but he's a biochemist by training. He's a professor of genetics at Harvard Medical School, professor of health sciences and technologies at Harvard MIT. He has appointments at the Broad Institute and the Wyss Institute for Biologically Inspired Engineering. I believe Wyss was Swiss, is he not? Which is a very exciting research center. He's the director of the Personal Genome Project. He's also involved in so many startups. His conflict of interest slide, I believe, has spilled over to 2 slides now. I think there's some 27 companies on there, but who's counting? One of the interesting things about George is that he maintains a list of the genes that could be edited to enhance human existence, either to prevent disease or enhance and extend our capabilities. And by the way, if you're interested in that link, you can find it at arep.med.harvard.edu/GMC, that's his lab, /protect.html, which I think is interesting framing, protection instead of enhancement, but we'll talk about that. So George has obviously been thinking about this for a very long time and very deeply and across multiple domains. He's imagined a couple of scenarios for us today about how his technology would play out among human populations. And so let's kick it off first by saying good morning to George and then asking him to outline his scenarios for us.
Hello, thank you, Jane. Can everyone hear me?
Yeah.
Okay, so I just want to frame this in terms of vaccines. There are 16 childhood vaccines that are among the cheapest and safest technologies in history. The ones that are on everybody's mind today are a 17th vaccine, which is Moderna, Pfizer, and AstraZeneca for COVID-19. These are as low as $2 a dose, and they are, I should point out, gene therapies, all three of them. And I think all vaccines could be classified as enhancements or augmentation relative to our ancestors who lived in fear of these 17 diseases and ones like it. So scenario one is editing. This is a kind of prevention, but now in the form of a cure. So scenario one is editing CCR5 gene, which is a receptor for HIV, to cure HIV-infected children postnatally. We should note that clinical trials for CCR5 for HIV-infected adults has already been approved by the FDA since 2009 for zinc fingers and recently for CRISPR. So scenario two is, again, editing CCR5 gene, but now in children to prevent HIV rather than to cure it. And it has the possible side effects of positively affecting COVID-19 and enhancing cognitive traits. This has been shown in mice, 3 different cognitive traits. This could be applied prenatally, not necessarily germline, but preventatively is the key thing. And fine-tuning could help avoid impact on West Nile and influenza and other immune functions that CCR5 participates in. FDA approval, as with all drugs, is necessary, and in this case would probably include a battery of immune, cardiovascular, and cognitive testing. Those are the 2 scenarios.
Great. Okay, thank you. So I do have a clarification. So the cognitive enhancement that would come with a gene edit to CCR5 is a byproduct. It's not a separate
it probably wouldn't even be labeled as such, but it is something that's been noticed in mice and would be something that probably in phase 4 people would be doing studies to see if it was significant or not.
Okay. Okay, so right away we've set up this place where it's very difficult to distinguish between what is a medical necessity and what would be considered an enhancement. So these are very slippery slopes. So what I'd like to do is get our panelists to weigh in on that. But before we go there, let's just take a little poll first of the audience. So if you could go to your device, go to slide.io, type in, when asked for your code, hashtag GESDA21. I guess it asks you to select room D, okay. And you look like you may be ready. So our first question is, do you support the idea of strengthening our immune system by permanently altering our DNA with new vaccines that have been proven to be safe and effective? Oh, okay. Okay. So we have, I think we have a lot of public health people in this community, which of course would be no big surprise. But we nevertheless have 30% of the audience who see some concern around that. So I think that's a pretty interesting outcome. On the next question then, do you think that genome engineering will intrinsically change us as humans? So George and I had this conversation. Fundamentally was the word that we were initially using. They said, well, you know, did trains and refrigerators and transistors fundamentally change us? Do jet airplanes and, you know, microscopes and telescopes change us? So what we're talking about is intrinsically change us as humans. So yes, a vast majority of you, 78%, believe that. And then the third would be, do you believe we will get to a point where genetic engineering will allow humans to acquire capabilities that we don't currently have, such as edited immune systems, enhanced cognitive abilities, and delayed or even reversed aging? Okay, so nobody is questioning the science per se. I don't know, 8% are doubting us. Okay, so that's kind of where everybody is. I'm not sure that we're going to change a lot of opinions with this panel, but I'm certainly curious to see if we need to poll again at the end. So since we have a renowned scientist from America here, we have a renowned scientist from Africa here as well, and I want to get him to weigh in on the scientific issues first. So Professor Ambroise Wankum is an MD and PhD. He's a medical geneticist. He's a professor in the Division of Human Genetics for the Faculty of Health Sciences at the University of Cape Town. Yes, exactly. And so, Ambroise, where do you go when you look at, you know, the idea of editing CCR5? As a geneticist, you must be very excited about its potential for preventing HIV and other viruses. But where do you come out on that?
Yeah, maybe to start, Jen, thanks very much for the invitation to share some of my thoughts on this panel, and thanks to other organizers. I would like to share my answer first for the queries that you put forward to the public. Do we need to permanently change our immune system? I would tend to say no. Do we think the gene editing can, I think the second question, can you remind me?
Yes.
So to that one, I say yes anyway. And the third, I said yes. But the reason why for the first, I say no. I'm thinking that I'm not even sure we have our priority right in terms of what we should edit. The second, I'm not sure we are genetically literate enough for a venture too quick in gene editing. And the third, I'm not sure we have addressed the question of equity in the level of gene editing. I will start with the first. As far as CCR5 may be a fascinating target because of the burden of HIV, and I live in a country where HIV is very high, I would tend to say it's not the first priority. HIV been discovered in the 90s, and we have efficient treatment that actually can work very well. But the first molecular disease, the first condition for which all humanity knows about genetics, translating genetics, is called sickle cell disease. And it's also the first molecular disease in terms of numbers. Every single year, we have 300,000 children that are born globally affected by sickle cell disease, and 80% of those kids are born in Africa. That makes our priority right. It's a single gene disorder. It's a disorder for which gene editing works already. At least 2 kids have been gene edited this year for sickle cell and cure for it. It's a condition for which, by making gene editing accessible to all the kids I see, we address at the same time the question of equity. The second around equity and around priority is who gene editing, who have the capacity of doing so. I live in Africa, in a country where we have probably 20 medical geneticists for 50 million. And I will have to address those questions. Who the workforce, is it equally shared globally to address that prospect? I believe opportunity is everywhere, and talent is everywhere, but opportunity is not. We have to make sure that the talent, we harness the talent everywhere where the priority are. The second point is that, are we genetically literate enough? The answer is no. We have 20,000 genes in our genome now. If we look in the OMIM database that we use as medical geneticists, only 25% of genes that we know are associated to disease condition. We have no idea what the 75% are there for. Are we genetically literate enough? The answer is no. We have no idea how epigenetic works and in gene interaction. We have no idea what the 95% of our genome that code for, that doesn't code for protein, what they're there for. So we should not be venture too quick in changing the genome without understanding the genome. Are we genetically literate enough? The answer is no, because ancient DNA that is introgressed in our genome from Neanderthal and from multiple humanoids that live in Africa with the 300,000 years with Africans of today, we have no idea how it works. Actually, we call them ghost DNA. And just to underline our ignorance. But let's imagine that those ghost DNA have an impact on the disease and on health. In a similar way, there are some strong hypotheses that the proportion of Neanderthal that we have in our DNA is responsible for the high susceptibility of COVID-19 in European and Asian. That means that we have work to do before we start editing our genome. And are we genetically literate enough to venture gene editing? No, because we have other technology that actually can allow cure of single gene disorder. And the COVID-19 vaccine have shown the way. And we have other mRNA, like messenger RNA, I mean that iRNA that can be used also to target a whole pathway of protein production without changing our genome. Thank you.
Great, wonderful. Okay, so very much concerned about moving too quickly. You see the possibilities, but you want to address the issues that are directly in front of us now.
Yes.
And make sure that we are doing this equitably.
Absolutely.
Okay, all right. So I want to go next to Effie Vajana, who unfortunately couldn't be with us today. She is zooming in from Zurich. Effie is a professor of bioethics at ETH Zurich. She's founder of the Health Ethics and Policy Lab, the Department of Health Sciences and Technology here in Switzerland. Effie, I think we've already got a sort of a bit of a polarization going, but we have enormous potential in these technologies. What could possibly go wrong? And how do we establish a baseline framework for thinking about how to move forward in advancing these technologies?
Thank you, Jane, and apologies I cannot be there in person. As you can hear, a virus has me down. So when I'm looking at George's examples, I'm quite biased actually about whether I would like on my DNA to avoid this misery, these miserable occasions. But beyond that, I want to pick up on something that actually it was your first comment after George's presentation, which was the question about cognitive enhancement and whether, you know, you almost sought for a clear boundary between the treatment of a disease and enhancement. And the first comment I wanted to make is this aspiration we have, or this need we have to hold firmly on the line between treating a disease or enhancing. At the same time, George started off by saying he thinks of immunization as enhancement. I kind of tend to agree with him. So from the kind of moral point of view, I think it's hard for me to see why it is actually relevant to make that distinction between disease treatment and enhancement. And that might be a controversial statement, but I would invite us to think a little bit about it by the following way, is that if we are trying to draw the line between the two, we're probably thinking of health as in its sense of physical health, perhaps even mental health, but we're not thinking of well-being. And if we think of well-being, and what we can do to improve that. I think the boundary between treating a disease and enhancing is, it gets very messy. So for me, from the kind of thinking about it from the moral point of view, I don't see the need for that firm line between the two. Again, and I think what's very important here, it's kind of, instead of thinking so much about the line between the two, is to think about safety and precision. And in the last comments, I think that's what we heard. Is our knowledge enough to allow us to develop these interventions in a way that they're safe and they're precise, they're doing the thing we want them to do instead of something else that we accidentally, that, you know, due to our lack of knowledge might not be what we wanted. So the question of safety and precision for me are the priority cases and the priority concerns. And of course, the sense of whether, you know, we're okay with our DNA being changed, it seems to me the question is, if our DNA changes over time beyond our control and without our understanding, we're okay with this. But if our DNA is to change because we took some control over it, then we're getting very anxious and worried. And that's sort of fair enough about the responsibility we're taking. So the first point is about the line and how we should draw its safety and precision. And the second is one that I think I find more challenging. Assuming that we are having those interventions prove to be safe, effective, precise, I think the problem we are going to run to is access and fairness. Who is going to access those good technologies and who is not going to do that? If you take the current, our current times, the example of the mRNA vaccines and who gets to have them and how many people do not get to have them, that tells you all you need to know about how this is going to play out. So the question now is, knowing that there is not going to be fair access, should that determine what we're choosing to do and whether we're progressing in a particular direction of inquiry of scientific discovery or not? It's a difficult question to answer, but I think we cannot ignore it because we know it's going to play out in a way that is not going to be fair or equitable. And the third point I'd like to, so next to that also, there is this broader question that's been put forward many times, and that is once we start making those choices about what we'd like to change or where we'd like to intervene, particularly in the space of what we call enhancement, that will cause also that would have consequences for many people who currently have those conditions or those traits or whatever we want to change. And we don't have a good mechanism of protecting those people from consequences, discrimination and other things. So that's something, again, we cannot ignore because we don't have a good way of fixing it. My last point is about knowing all this, how do we really decide essentially about which version of progress we sign up to, the scientific progress, the societal progress, and how do we revise our notion of progress as we move on? Because I think we do revise it. If we look over the hundreds of years, we're constantly trying to improve the human condition with all means we have. We want to reduce human suffering. We want to improve well-being, but we revise constantly the ways in which we do that and its meaning. So the question again is, how do we do that in a manner that allows all voices to be heard and all disagreements that you mentioned in the beginning to be deliberated? My worry here is that we have not come up with a good format and a good formula of doing this. Very often, and Catherine can attest to this, in our circles of bioethics, we end up these conversations by making a call to public engagement, to stakeholder engagement. We think that's a solution to many of these difficult conversations. But my feeling is that it's wishful thinking. We're trying to do this. It's a very difficult thing to do, but we're not really having made incredible progress in this formula of global deliberation. And I'm not talking of just polling people. I'm also in the business of asking people's opinions and studying the results, but I'm talking about the actual deliberative process so we can figure out how far we can go. So I really welcome the initiative of GESDA. I think it's an opportunity to lead us to this global dialogue we need. But again, it's about deliberating about our disagreements here and our opinions, not simply stating them. Thank you.
Thank you so much. It's absolutely true. There's an enormous role for GESDA to play here. If for no other reason than to just bring the various parties together in one room, create a common language and a sort of baseline of knowledge, and then frame the questions and the issues. It is astonishing to me that this many years after the development of CRISPR, that's still not really happened. We do have, obviously, the scientists concerned about the development of CRISPR are talking amongst themselves. And since Silomar, of course, have agreed that germline editing is unacceptable still in humans. But we also, Jennifer Doudna has CRISPR-Con, in which she brings together scientists with food people and with policymakers and puts them together. But, we really are missing all of the stakeholders, including private industry, to come together and have these conversations. So definitely a work to do on GESDA's to-do list. But I think what would be helpful is to talk to Catherine Littler next, because she's working on the policy side. And Catherine is the co-lead of the Global Health Ethics and Government Unit and the Research for Health Department of the Science Division of the World Health Organization. You NGO people have the longest titles. It's crazy. I don't know where to pause and put commas or breathe. You'll have to coach me on this. With a focus on ethics, governance, and policy. Catherine was, I believe, the editor of this amazing human genome editing position paper. There's a much longer document on the recommendations. Yeah. And if you look at the WHO's site, you will find a huge number of documents that all got generated and published, I gather, in July.
Yes.
So obviously you've been very busy. We're delighted you can join us, but there was one sentence in particular that really resonated with me, and it was so well written. I wish I'd written it myself, so I'll just read it. We have to consider the balance between benefit and harm, safety and speed, innovation and access. So with that in mind, Catherine, what is the current state of affairs in terms of regulation, in terms of policy recommendations, in terms of how the WHO is grappling with the power of this technology?
So the simple answer is there is no simple answer. I think what's really interesting about this discussion is how gray it is, how gray the areas are, whether you're talking about the language, which technologies you'd focus on, or where we're going. There is a plethora of governance mechanisms out there, depending on whether you're talking about somatic or hereditary or germline. It really depends. And I think we're at very differing stages all over the globe in terms of governance and oversight. And we have very different starting points of what we think is acceptable or where we are starting from. And I think Ambrose alerted us to that. George is starting from a very different starting point of what's there from where Ambrose is. So I think that raises really interesting issues. There's also, as Effie picked up on, and what we're picking up on with these types of technology, issues of certainty. There's a lot of science that needs to be done still. And I think actually there's interesting parallels at the moment with COVID. One of the things after working on the human genome work for 2.5 years, the committee worked on it, an 18-strong committee, and they came out with the position paper, a governance framework for human genome editing, which is really a toolkit, and a series of recommendations, which is looking at some of the systemic challenges. But what really struck me and is still striking me in these conversations are the gaps and what we need to focus on. Something that's really important, I think, if we're going to realize the potential benefits of these technologies is preparedness. We talk about epidemic preparedness, but we should be talking about preparedness for genome and emerging technologies. And when I define preparedness, I'm not talking about just the science. I'm really talking about the governance, because I think governance really is not the panacea, but it's what will help us address a lot of the challenges. So the committee really focused on the governance issues, noting that the starting points are very different. It's why it developed a governance framework with scenarios, which actually are relevant to the ones that George raised, looking at the questions that you should be answering, looking at the governance mechanisms. I think this is really important in emerging technology. You cannot have a single governance mechanism. No one player is going to be responsible for it. No one player is going to be making the decisions about what we do, what technology we do. There is a democratization going on, but that doesn't mean everything is available to these technologies. So I think we have to be cognizant that there are a lot of players in this space. A lot of players have responsibility. Governments have responsibility for putting in place regulation and legislation. But as we know, that doesn't always keep pace. Research funders have responsibilities. UN organizations have responsibilities for looking at all sorts of international layers. So I think there's some really interesting challenges here in terms of governance that having these types of discussions bring to the fore. I want to raise the interesting question around language. It's hard to have a debate when we don't have agreement on language, and enhancement is a great one because we just saw everyone's defining it differently. So, and then how do you have a conversation with the general public when I think they might have a much more black and white perspective of what enhancement is? So I think even getting some consensus within us and our communities about how we use language. Effie raised a great point about engagement, and she's right. I've been in policy for 20 years. When I first got in research policy, you didn't have engagement as a central discussion on a platform like this. This would not be included. Now it's everywhere. It's everywhere. And Effie's right, we don't know how to do it effectively, but that doesn't mean we shouldn't do it. It just means we have to be better at deliberative democracy and thinking about what it needs to look like. But I think engagement is a big factor. When I talk about engagement, it's not just publics, it's policymakers. Understanding these technologies, the risks, the potential benefits, the potential risks is really important. And I think there needs to be an education process that goes along with policymakers if we're going to, and with publics, if we're going to realize the benefits of these types of technologies. And I want to finish on the question of equity, because the Human Genome Editing Committee that WHO put together really did focus on equity issues. And for me, that's part of this preparedness agenda. When people think of horizon scanning in the science, and these types of meetings are about that, where are we going to be in 5, 10, 25 years? They often just think about it in terms of where's the science going to take us? But we should be thinking when we get an idea, how can we make that idea accessible across the globe? Like, what would we need to do around the science to make that available? How do we bring the cost down? What are we thinking about in terms of patenting? How do we bring these sorts of discussions in really early? And I think this is where governance does play a really key role, but I'll stop there.
Great. Okay, that's a lot. All right, well, I do think, since almost everybody mentioned this, that I would like to get a conversation going around this concept of enhancement versus treatment. Effie suggested, perhaps provocatively, that that's not a useful distinction. So I'm just curious what the rest of you think. And I'd like to start with George, because you gave us scenarios that blur those lines. Are they useful? Is that a useful distinction or not? Is it a distinction we can even make?
I think almost all powerful and popular technologies are enhancements, whether they're cars or books or computers, et cetera. I think what's more significant here is whether they're reversible or not. So if you try to reverse, say, the telephone revolution, that would be very politically very difficult to do. So in a way, that's irreversible, at least so far. I also think that when you talk about equity, which every single one of us talked about, was my first comment was, how do we get equitable distribution? The most equitable distribution comes from the number of people affected. So viral diseases affect 8 billion of us. 6 million a year die from unvaccinated viral diseases. In other words, we get 16 or more vaccines, but there are a few that aren't. And so when we talk about a disease like sickle cell, which, by the way, one of my companies produced one of the gene therapies that Professor Wankman talked about, 300,000 people is not going to be as equitably distributed as something that affects 8 billion. It's going to be expensive. Almost all the rare disease gene therapies are in the hundreds of thousands to $1 million, while the vaccines are closer to $2 per person.
So if I'm hearing you, what you're essentially saying is that perhaps if we moved forward with things that alter our DNA, that change it for a lot of people at the same time, that's more interesting or equitable or desirable at this stage of things than very expensive rare disease treatments.
I think we should try to do both, but there are alternatives for rare diseases that include genetic counseling, which are hundreds of dollars rather than hundreds of thousands. But for vaccines,
the counseling to prevent the embryo from forming.
Preventative, yeah. But for vaccines, these are definitely augmentation and enhancement, to your point, and they are equitably distributed. In fact, they're the only technology that's ever been equitably distributed, because all 8 billion of us are not at risk for smallpox the way our ancestors were, and they don't need to pay a penny for that. It's completely free for now until the foreseeable future, because it's extinct. And I think many of other human diseases can be made extinct, not all of them, but that's the way to get equity.
Ambroise, you were talking about sickle cell anemia. What percentage of Africans are at risk for that? And would that be a gene edit that you could see widely deploying?
Yeah, probably I will start by commenting on George's concept of equity that aligned to the numbers. I like the concept that when it affects more people, probably it's more equitable to target it as a priority. But I'll still come back to the concept of genetic and genetics disease. When we speak to equity here, I'm speaking as a geneticist, as a medical geneticist. My number one priority is number one recessive condition of humankind, and there is only one, is sickle cell disease. My number one priority is hemoglobinopathy that affects 7% of the world population in terms of carrier. And if we also want to speak about equity, I will also come back to the concept that we are not genetically literate enough to manipulate our genome. There is a concept in Akan tribe or language. It's a group in Ghana, the concept of the power of Sankofa. The Sankofa is a symbol of a bird turning back and picking an egg on his back. And actually, Sankofa speak about the past, but the egg is to fertilize the future. I believe that we have to go in the past of our genome to understand how actually our genome combat infection by selection, instead of trying to creating a new ways through editing. And I will speak to a few examples. Sickle cell is one. The selection of the sickle mutation was a fight against malaria. As we know that the selection of OPL1 was a fight against trypanosomes. So we have numerous of those examples that our own genome naturally created in terms of therapy and resistance to the environment that we have not yet even explored. Because if we know how this happened, maybe it will allow us a better way, an acceptable way to manipulate our genome in a more understandable way. I will again speak to the power of Sankofa again. There is only 4 conditions over the human history or evolution that have changed our genome. The first is ecology, the pressure of our genome by the environment, and selection in our genome of things that allow us to resist to our environment. The second is ancestry. The admixture and migration of humans have created other variation. And the third, of course, will be behavior. In area of the world where consanguinity is acceptable, you have that type of variation that become very prevalent. And we are already now adding an intervention, which is the fourth, through editing, that potentially can change our genome while we have not yet mastered the first 3 conditions that get our genome evolved over time. Equity will mean to my view, that all part of the world have access to the technology, all part of the world have the human resources in terms of workforce that can equitably actually implement the technology. Equity will mean access to funding to investigate priority that are relevant to all part of the world. Equity will mean access to platform like this one so that all voices have been heard. It will not be not necessarily about epidemiology.
That was fabulous. And this is why we bring together people from different parts of the world, is to thank you, Olivier, for putting together such an interesting panel. So Western-centric perspectives make it very difficult to talk about what is our highest good. And I think perhaps our highest good is, from your point of view, understanding where we are better before plunging headlong into the future. But that's really interesting. And, you know, Catherine, you have this really wonderful framework, you know, back to this sort of enhancement versus treatment question. You had a really useful framework here in which you identified the 5 different challenges of sort of the postnatal somatic human genome editing, prenatal, which would be in utero, you know, heritable, human epigenetic editing, and enhancement. Do you try to grapple with the concept of enhancement versus treatment in your recommendations?
We do. We do it via posing questions, as in what are the questions you need to consider if you're going to take forward an enhancement. Because I think Effie's point is important here. Maybe the difference between enhancement and what we're calling non-enhancement isn't the issue here. Maybe here is partly the intention, partly the equity issues, which I'm just going to say isn't just a numbers game. I think that's it's an important point, the numbers, but it isn't just a numbers game. So I think intent's also important. One of the key things with enhancements, depending on how you define it also, is, and with equity issues, are you exacerbating existing social or economic inequalities by proceeding with them? So I think in order to, whether you think an enhancement is acceptable, how you define it, should be done with I think we look at rules of play, I'm going to call them, questions you should answer yourself, mechanisms and means if you're making these decisions. Because I think there are valid points. It's not the language that's important here. It's often the purpose and the benefit and all those other things that are downstream that are the critical points.
So coming from Silicon Valley and being very familiar with the innovation curve, and familiar with how very, very expensive technologies are purchased by early adopters, which then starts to form a market, which then creates an opportunity for the prices to come down. How do you square that kind of thinking with the idea that we can only deploy these technologies if they are available to everyone at once? I mean, aren't there just going to be people who will not have access at the beginning, and perhaps it's a less perfect experience, a less perfect outcome. So they may be at greater risk, but they smooth the path for the people who come after them? Or is this a completely wrong model to bring to genome editing conversation?
Yeah, probably one of the questions is, how do we define excellence? Is excellence means having the most powerful, at least in terms of research and innovation, technology that actually transform the life of a few, of having the most powerful discovery that transform the lives of so many. If we come back to COVID-19 once again have shown how the half in terms of technology would actually shift the global agenda to some extent. But at the same time, if you do not have that technology accessible in terms of vaccine availability, you are harming yourself because you cannot live in isolation in a world that is globalized. I do think excellence will have to be redefined by not only the impact on a few, but the impact on masses.
Absolutely. So I just want to stop at this point and say we've got about 15 minutes left. I'd very much like to hear what people in the audience are thinking about. If you guys have some questions, would you please send them in? Hopefully, I only have one screen. I've had two screens in the past, but hopefully they'll pop up here and we can start to see them. Okay. And I will just remind us of what our session objectives are, because I don't think I read them at the beginning. We want to showcase the big questions around the future of human genome editing by identifying the key risks, challenges, and opportunities. We want to illustrate the policies currently developed around the world to deal with those issues and where new policies and actions are needed, which of course is GESDA looking for the best value they can offer. And then to highlight the impact on each participant as an individual. What does it have to do with me and our way of living together in society? So Catherine, I'd like to come back to you on diplomacy and the role that it has to play. Every country in the United Nations is going to come in with a different perspective on this. And so, you know, is the World Health Organization going to have to adjudicate between, you know, America and its Silicon Valley, you know, capitalist mindset and, you know, South Africa or some other country that may have a completely fundamentally different agenda? Who's going to adjudicate these conflicting interests?
So this came up a lot in the committee's deliberations. It came up in overall discussions about when we should proceed and when we shouldn't, and should we be putting in place international mechanisms? Where do voices like WHO come in? This came in a lot, and I think what the committee settled on is there is a lot of responsibility in this place, which is where you need this brokerage. So I would say science diplomacy going forward in emerging technologies will be critical because there are a lot of different players with responsibilities. And you might not get global consensus on everything, but it's good to work towards that as much as possible. Acknowledging that you have different cultural contexts, different starting points, but there should be some form of reciprocity or acknowledgement of different types of processes. So I think you do need organizations like WHO where they have strength in the health sector. But for example, the committee advised that the UN set up a mechanism on engagement for new technologies, that not being just WHO, because it circumvents not just health, it goes beyond. But I think the interdisciplinarity of this area, as well as the fact you can't just talk about human genome editing, you'll probably also, and Effie will know this, you're going to talk about AI in relation to human genome editing. There's an intersectionality of all these technologies, which means that brokerage and thinking with that big picture lens is only going to become more and more important. And that's what the committee kind of focused on. It also focused on a very practical governance framework. What are the rules of play? How do you take the decisions in different contexts? But then it looked at some of these huge systemic issues surrounding emerging technologies and who are the players that should be in. So I really think science diplomacy is the only way that we're going to proceed, because you've got to get so many different parties to speak to them, to each other. And there's accountability and responsibility, and even working out, not everyone's acknowledging where their responsibility is or their accountability is. So having these discussions, I think, is critically important.
So I'd like to go to you, and then I'll start taking, yeah, there's great questions. Okay, but one more, and that's for Effie. Will there be one consensus? I mean, the idea seems preposterous to me that there could be, but I'm curious what you have to say on the topic.
Well, I think it depends on what. There might be consensus on certain things, but sort of consensus on everything, I don't think we're going to have. With the conversation between George and Ambroise was also about priorities, and different contexts will have different priorities. And we cannot just say that that's not true. That's the reality. But what I think is important is back to what Catherine was saying. We need this place where we can understand the different priorities and what we can do for each other in a more solidarious kind of way, because we have to acknowledge that we have these global problems despite our national or kind of, regional priorities. I think that's where I hope we can get more of the consensus, and that's why fora like the WHO's or GESDA can help us do that. But getting consensus on everything about all things around gene editing, I think we should be realistic and say upfront that that's not possible. If we were, though, to get agreements on some of the important things that matter, even in the process, process of setting priorities, forget that we might end up with different priorities, but the process of doing that, I think that would be an enormous achievement in my view.
All right, well, you can't see this, but there's a lot of people here who want to ask some questions. I'm going immediately to Olivier Desibourg.
No, these are online questions, actually.
Oh.
I relay the online questions to you.
Oh, okay. Should we take some in-room
let's
questions
take one in the room
first?
and then I'll take the online ones.
All right, great.
Hi, Conrad. I was just wondering regarding innovation versus access. I think you mentioned that Catherine wrote this in her WHO report, and the practical reality is such that we're currently unclear about how to allocate our resources, but what at the moment is the resource allocation? Like, are we doing 50-50 at the WHO, focused on access versus innovation? How do you guys think about the trade-offs here? Because focusing more on equitable access probably also slows down innovation. I mean, it's not always that clear, but there must be some thinking here. And I was wondering about the practical reality.
Yeah, so we've within the science division, one of the partner teams in my area has set up a horizon scanning mechanism exactly to think about this. How do you balance the trade-offs? How do you do this without slowing down innovation? And how do you make the governance and factor in all these other, as Effie pointed out, even if you think about doing the engagement, that's going to slow you down so much. And the governance, if you don't do the governance right, I don't think we have the magic bullet answer. I don't think anyone does. And we definitely do not want to slow down potential innovation for health. And we're working definitely at the country level where they're implementing this to see what's going on the ground. But I don't have that answer. I can just say we are working on it. It's definitely in our horizon scanning work.
So these are more scientific questions. I'll group them to George and also Ambrose. So the first one is, George, are you in the opinion that mRNA vaccines and similar therapies will be on the first line, on the fast lane, while gene editing therapies will wait for some time, and why? This is the first one. And the similar one is, so to Ambroise and George, where is the line of genetic literacy? Some would argue we know very little about some drugs that are used currently. And the last one, a detailed question maybe to George. George, you talk about reversibility. Will gene editing and CRISPR be reversible in the future? So let's start with George, maybe.
Okay, so are they on the fast lane? I actually, to Professor Wonkam's statement about prioritization, I think that sickle cell was a higher priority for companies I've been involved in than more common diseases. So when COVID came along, though, then additional priority was added. They didn't stop working on sickle cell. Now, how fast they get through clinical trials depends on the risk versus benefit. So I can't predict. I mean, clearly, COVID-19 went through clinical trials faster than did the sickle cell, but the sickle cell did get the high priority for being initiated first. And hopefully it will go very quickly as well. In terms of literacy, I should point out that consensus, literacy and consensus, is I think we can achieve consensus on some very important topics, as we did for smallpox. And I should point out, we did not wait for smallpox vaccination for full understanding of all the genes, or even understanding of virology or immunology. We started vaccinating before we could see a virus, before we even knew there were viruses, and before we knew anything about immunology. So I'm not recommending that we act on ignorance. I'm just saying that occasionally we can reach consensus without full understanding. And in terms of reversibility, I think that editing is definitely reversible. I'm not advocating editing in our genomes so much as editing changing our genetics. So it could be messenger RNA, which is perceived as being temporary. The AstraZeneca vaccine is actually an adenovirus carrying a double-stranded DNA. So it has nothing to do whether it's RNA or not. I think if we can go forward, we can also go backward. But I don't think there's an argument, really strong argument for doing the germline, since we can do so much after babies are born, after we're adults even. And in fact, since most of us are adults, not newborns, 8 billion of us are basically beyond the newborn stage, there's going to be a lot more emphasis, priority, and success with drugs that are aimed at newborn and beyond than anything in the germline. There's just not a strong argument for doing germline. So that's in terms of reversibility. I think we are getting quite good at, if we can go forward, going backward. But we should not be naive about it. This is exactly the sort of thing the FDA, the EMA, and agencies around the world are tasked with carefully evaluating the safety and efficacy.
Great, let's take another question. Just give us your name, please. And
Philip McDonough. My name is Philip McDonough. I'm a former Irish ambassador, and I've started a centre to explore values in diplomacy in Dublin. I just wonder, should we think of religion in this discussion? You know, most world religions have some sense of life as being a gift, which should be taken with in a certain spirit. So that, should we really ask ourselves whether we're interested in supporting life or in some way taking control ourselves over a mystery that's bigger than us? You know, is that way of thinking not an important part of the mix?
Mm-hmm. Well, I think that speaks immediately to fundamental cultural differences that could trump health concerns or global population issues or
I wouldn't wish to be put into that box. You know? I mean, the key word is justice.
Yes. Point taken. Did you want to say anything about that, Effie?
Yeah, maybe quickly that, well, I understand the point about religion and values, but in terms of taking control, I think over the years, we're trying to take control over our lives in a number of ways, and change and reduce our physical vulnerabilities. It is not the first time. Of course, in this form, it's a newer form and it's perhaps more invasive than others, but it is not new to humanity that we're trying to control the environment we live in ourselves. I grew up in a place where my parents had to test for thalassemia before they would get married or have children because that was prevalent in that region. And the story I grew up with is that if they were found to be carriers, they wouldn't have children. I wouldn't have been born if one of them had. So they tried to take some control, of course, not in the way that gene editing would do, but in some other form. So I think I understand the question that was put, but we should not reject also the fact that embedded in these values is the fact that we try to control our environment and ourselves in order to improve our condition over the years.
Excellent. Do we have time for one more question? Okay. Do you want to take another? Yeah. Do we have another question? There was somebody in the second or third row. No. Okay. Well, this has been an incredibly interesting conversation. I think we've found some real points of tension on how to move forward between going forward in ways that can touch the maximum number of people with the greatest benefit to humanity. But also trying to understand what exactly is the greatest benefit to humanity, how that differs from one culture to the next. Do we need to think more about what we don't know and explore that, or move forward into areas that we think we understand some of and can make progress on? What are the important distinctions that we need to make? What are the language? Perhaps how is our language not yet adapted to a consensus that we're all on the same page. How do we prepare for future pandemics, obviously, is a big one. What is the role of diplomacy and policy, and how will this evolve? And I do like the idea that equity is not just a numbers game. And so how can we move forward without a full understanding, both in terms of the science and in terms of the policy? It's very difficult to write a law when you don't know how things are going to go. So I think everybody here probably agrees that moving forward with human germline editing isn't something we want to do at this point in time, because there's so much important science that still needs to be done before we get there, and we can address a lot of this afterwards. So I hope that if you have any additional thoughts or questions, that you will participate in GESDA's radar, which you can find on the website. It is not just a really interesting timeline for how all of these different technologies will play out over 5, 10, and 25 years. It's also an interactive platform. And so your thoughts, your questions, some of which could be answered, some of which haven't been asked yet, are all welcome. And it's an evolving platform. So it's an evolving conversation. And I hope we all look forward to engaging in it further. So thank you, George. Thank you, Effie, for zooming in with us. Thank you, Ambroise, for coming all the way from South Africa, and Catherine for coming down the street. Thank you.